Hello, this video is talk about the peroxisomes. Since we have learned about the chloroplast and mitochondria, so now we go forward to peroxisome. Peroxisomes is a single membrane bound organelle containing oxidative enzymes such as catalase. This is different to lysosome. Lysosome containing hydrolytic enzyme, whereas peroxisomes containing oxidative enzymes. So here is how the peroxisome is formed. From here you can see the vesicles spark from the endoplasmic reticulum, they fill with each other. So to form a premature peroxisomes. Then several premature peroxisomes fuse together to form a mature peroxisomes. So these peroxisomes is able to divide into many smaller peroxisomes. So this is different to the lysosome, whereas the lysosome unable to be divided. Okay, peroxisomes, they can grow larger, as I told you just now, okay, by incorporating protein synthesized by free ribosome in the cytosol. So once the free ribosome, they synthesize a polypeptide. So this polypeptide is imported into the peroxisomes. Then the peroxisome becomes grow larger or becomes bigger. So when they reach the certain size, so the peroxisomes will divide into more new peroxisome. So enzymes in the peroxisomes, they are oxidative enzyme in which they carry out or conduct oxidations reactions or oxidative reactions, in which the hydrogen is transferred from various compounds to oxygen, result in hydrogen peroxide. So as you can see here, so any compounds, R is represents any compounds, so they containing the hydrogen's ion and the hydrogen's atoms. So they react with oxygen. So after that, the product will be, one of the product will be hydrogen peroxide. Then a variety of substrates are broken down by such oxidative reactions in peroxisome, so including uric acid, amino acid, and fatty acids. Peroxisomes get their name from the fact that during this oxidation or oxidative reactions, they produce hydrogen peroxide. So that's why they get the name peroxisome. Then the hydrogen peroxide actually is used to detoxify certain compounds. However, they would damage other membrane if the cell if it were to escape from the peroxisomes. So that's why in the peroxisome there is another enzyme which is known as catalase, which rapidly split the excess hydrogen peroxide to water and oxygen, rendering it harmless. Okay. Then in the liver, peroxisomes are also involved in the synthesis of bile acid, which are derived from cholesterol. And in human liver and kidney cells, the peroxisomes detoxify certain toxic compounds, including ethanol. Okay, for example, can you look at these equations? The ethanol is catalyzed by catalase, okay, and by using the hydrogen peroxide, so to form the acetaldehyde and water, which is less toxic. So in leaf, plant leaf, the peroxisomes can carry out reactions that is opposite to photosynthesis. As you know, photosynthesis use carbon dioxide and water to synthesize glucose, whereas photorespiration is a process to use oxygen. Okay, to use oxygen in the presence of light to produce the carbon dioxide, but no glucose is produced as in the cellular respirations. You will study this photorespiration in detail in the chapter of photosynthesis. So here I just let you know, so during the photorespirations, so whereas the peroxisome is involved. Okay. Okay, in plant, the germinating seed, specialized peroxisome we call the glyoxysomes. Okay, glyoxysome. They contain enzymes that can convert the stored fat into sugar. Okay, convert fat into sugar, the glucose. Then the sugar are used up by the young plant as an energy source and as a component to synthesize other compounds, for example, the polysaccharide. Then because animal cells, they lack of the glyoxysomes, so animal cells are unable to convert the fatty acid or fat into sugar by this way. Okay, so this is the peroxisomes. Normally, the structure is quite similar to lysosome, but they are different. Okay, because of the enzymes inside the organelles and also the reactions and the way how they form. So this is the peroxisomes. And thank you for your listening. Bye.